Today, we're diving into some really high-stakes genetic detective work. We're going to look at how scientists are using one of the most powerful tools out there to crack medical mysteries that have left doctors stumped for years, and in the process, giving some real answers and hope to families who have been on a very long, very difficult journey. So let's start with a question. Imagine you're a detective on a tough case. You've followed every standard procedure, you've checked all the usual clues, but every single lead has gone cold. You're at a dead end. Well, this is exactly the situation that doctors and families face when they're dealing with rare genetic disorders. So what do you do next when the trail runs cold? The story we're exploring today centers on 260 families. Every one of these families has a child with an intellectual or developmental delay, and even after all kinds of testing, nobody knows why. They've been on what's often called a diagnostic odyssey, which is just a painful, years-long search for a name, for a reason, for their child's condition. Now, the first investigation into these cases used a pretty standard genetic tool. But as we're about to find out, that tool had a huge blind spot, and it left the most important clues completely hidden, right there in plain sight. The first tool they used is called exome sequencing, or ES for short. Okay, think of your entire genome as this massive instruction manual for how to build and run your body. Exome sequencing is kind of like a detective who's only allowed to read about 1% of that manual, just the key chapters that have the instructions for making proteins. But what happens if the critical clue, the one that solves the whole mystery, isn't in those chapters? And this really shows you the scale of the problem. That 1% of DNA that was initially searched, it's just the tip of the iceberg. That first investigation completely missed the other 99% of the genetic code. That is a massive, unexplored territory where the real answers could be hiding. So with that first investigation pretty much stalled, it was time to call in a specialist, a new detective with the power to search the entire crime scene. And this is where we meet genome sequencing, or GS. This comparison really nails the difference. If exome sequencing is like reading just the key chapters, genome sequencing is like reading the entire book, cover to cover, including the introduction, the tiny footnotes, the appendices, everything. It doesn't miss a single letter, so you get the whole complete picture. So here was the new plan of attack. Researchers took those 260 unsolved cases and brought in the full power of genome sequencing. Their goal was simple: hunt for clues that were hidden in that other 99% of the DNA, in those non-coding regions, and even in the very structure of the chromosomes. They were hoping to find answers that were basically invisible before. Okay, so what did this new, more powerful detective actually find? This is where the story gets really, really cool. Genome sequencing started turning up 3 very specific types of hidden clues that had been completely missed the first time around. All right, the first big breakthrough came from finding something called structural variants. Now, these aren't just single-letter typos in the genetic code. No. Imagine entire pages or even paragraphs of the instruction manual being accidentally copied or deleted or moved to a totally wrong chapter. These are big, disruptive changes that exome sequencing just isn't built to see. And here's a real-world example. In one patient, genome sequencing found a huge structural error. A large chunk of their genetic code had been duplicated. This wasn't a small mistake, it was a major rearrangement. And this finding was so important that it actually led the researchers to propose an entirely new syndrome, giving this family an answer that was just impossible to find before. The second type of clue was found in that giant, unexplored 99% of the genome. For a long time, scientists kind of thought this non-coding DNA was just junk. But oh man, we now know it is full of critical instructions. Think of them like the on-off switches and dimmer dials that control when our genes get used and how much. And this is a perfect example of what I mean. In another case, GS found a small deletion in one of those non-coding areas, basically a broken control switch. Now, this switch wasn't in a gene itself, so exome sequencing completely missed it. But breaking that switch caused a neighboring gene to go into overdrive, which is what they believe caused the patient's severe condition. And finally, we have the third type of clue, something called a repeat expansion. You can almost think of this as a kind of genetic stutter. It's when a short sequence of DNA letters, like CCG, just gets repeated over and over and over again, way more times than it's supposed to. And that just wreaks havoc on the gene's function.
And that's exactly what they found in another patient. The deeper analysis from GS uncovered a rare stutter right next to a really critical gene for development. This massive repeat is thought to have basically silenced that gene, turning it off when it should have been on. It was another smoking gun that only a full genome analysis could have ever found. So, after all of this amazing detective work, finding these rearranged blueprints, faulty switches, and genetic stutters, what was the final verdict? For these families who had been searching for so long, what did these new clues actually mean for them?
The bottom line is this. For cases that were completely unsolvable before, genome sequencing provided a definitive or a really strong potential answer in 10.5% of them. That means more than 1 in 10 families finally got an answer where there was absolutely nothing before.
Let's break that down a bit. Almost 7.5% of those previously unsolved cases got a definitive or very likely diagnosis. Another 3% got some very strong clues pointing to a cause. And while yes, a lot of cases are still a mystery, this is a huge step forward.
And why does getting a diagnosis matter so much? Well, for one, it can finally end that painful odyssey. It gives families an answer. But it does so much more. It can change how a patient is cared for, and sometimes it can even point towards new treatments. In fact, for 2 patients in this study, their new diagnoses actually open the door to existing or potential therapies. So to sum it all up, the superpowers of genome sequencing really come from its ability to see the whole picture. It finds those big structural rearrangements, it analyzes those critical non-coding regions that act as switches, and it catches the genetic stutters that other methods just can't see. It's a fundamental shift in how we look at our own biology. And this is really just the beginning. We've managed to crack a few more incredibly tough cases by reading the entire book of life, not just a few chapters. It just makes you wonder, as this technology gets even better, what other secrets are just sitting there waiting to be found in our DNA?